Welcome, everybody. My name's Kim Killian, and we're excited to have Dr. Cooke launch or kick off our 2021 regional webinars. Dr. Cooke comes from us from Houston Methodist, and today he's going to be talking about democratizing RNA nanotherapeutics. Just quickly, I wanted to do an introduction introducing our North America West team. So in the Northwest, we have our regional sales director, Angel Galarza, who is based in San Francisco, along with Ian Villamagna, who is his field application scientist, also based in San Francisco. And the Southwest team is comprised of myself. I'm Kim Killian, again, based in San Diego. I'm the regional sales manager, and I work in tandem with my field application scientist via WIN. As a reminder, PNI headquarters are based in Vancouver, and we also have sales support globally. Okay, and then just finally, we do have many quarterly events planned from now till the end of March. We have our journal club, which will be February 12th. It's on CRISPR-Cas9. It's hosted by Dr. Wynn. We also have webinars at the end of February. So if you want to register, we'll send you a link after the event, and you can sign up for the next journal club, February 12th. We have our journal clubs the second Friday of every month, and we have our webinars the last Wednesday of every month. So now I'd like to hand this over to Dr. Cooke. Thank you again for joining us, Dr. Cooke, and we're excited to hear what you have to present for us today.
Well, thanks so much for the introduction, Kimberly, and the opportunity to speak today about this topic that is of great interest to all of us. This is more of a conceptual talk, and I would love to get people's comments during the talk or immediately afterwards. I think it's meant to stimulate discussion and thought. As Kimberly mentioned, I'm here in Houston in the Texas Medical Center, and I am the chair of the Department of Cardiovascular Sciences and director of our program in RNA Therapeutics. The medical director, Roman Sokoverson, is the scientific director of our program. And I'd like to tell you about this idea that we have about democratizing RNA therapeutics. But before I start on that, let me just tell you a little bit about who we are. A little slow to advance here. There we go. We're in the Texas Medical Center, based in Houston. The Texas Medical Center is the largest medical center in the world. There's actually close to 60 research and medical entities with over 100,000 employees. It's like a small city in the city of Houston. We have a very rich and diverse scientific community. We have a dense network of collaboration with our colleagues at Baylor, at Rice, at UT Health, at Texas A&M. So it's a wonderful place to be doing science, translational science, clinical research. Our hospital in the Texas Medical Center is ranked #1 in Texas by US News and World Report, #20 in the US. We have a top ranking in 11 of our specialties. And it's a big place. We have the mothership in the Texas Medical Center, and then we have 7 outlying hospitals ringing Houston. We have about 1 million patient encounters a year, and almost 5,000 physicians, 600 faculty in our group. What's shown on the left is our beautiful new research institute, which was built in 2010. And just has wonderful facilities, including GMP facilities that I'm going to introduce you to for the generation of therapeutic clinical grade RNA. Now, what do I mean by democratization of RNA therapeutics? What I mean by this is that I'll show you evidence that small entities, academic research groups, small companies, can bring innovative and personalized RNA therapies all the way to the clinic. RNA therapeutics will not solely be the province of big pharma and biotech. So everyone on this call that has an interesting idea, it is possible for you to get that therapeutic idea to the clinic. And I'm going to show you how that can be done. And the reason for this is that RNA innovation is rapid and relatively low cost. Much of the development work can be outsourced. We'll talk about that. We have facilities and expertise to help small companies and academic groups to get their work to the clinic. And yet you maintain control and ownership of the product and the direction of the research program and product development program. So academic research groups and small companies with a great idea and intellectual property can advance to clinical trials without the burden of a large corporate partner or losing control to venture capital. I do think, we may be the first hospital-based center for RNA therapeutics, but we won't be the last, because hospital-based centers for RNA therapeutics can outcompete big pharma in some areas, in some niche areas like personalized RNA therapies for cancer. And we'll talk briefly about that. Well, let me begin by saying, of course, recognizing this group knows that there's a great diversity of RNA species. There's tRNA, rRNA, microRNA, long non-coding RNA, and a number of other RNA forms, including circular RNA, in addition to messenger RNA. And but we're really going to be focusing on messenger RNA for this talk. And the reason to focus on messenger RNA is, as you know, it's a really rapidly emerging industry. The emergence of RNA therapeutics reminds me of what happened in Silicon Valley in the 70s when biotech, company Genentech made its first recombinant protein. So recombinant protein technology back in the 70s and 80s spawned the growth of Silicon Valley's biotech industry. And this is emerging even more rapidly and impressively. So the big three in the RNA therapeutics, messenger RNA therapeutics arena are CureVac, which doesn't yet have a product on the market, but is valued yesterday at $18.6 billion. BioNTech, which has their first product on the market, you guys well know, the RNA vaccine against COVID. They're 26.4 billion, and Moderna 52 billion, also with an RNA vaccine that's been approved for emergency use authorization by the FDA. So, I think we understand why the valuations are so high in these companies, because in part, there's a potential to disrupt the $200 billion therapeutic recombinant proteins industry. RNA is much faster in terms of its development, and it's much faster in getting to the clinic. As you know, it's essentially biological software, so you can write the code and make the product rather quickly and start testing it. And it's much simpler than generating therapeutic recombinant proteins, which have to be folded correctly and perhaps post-translationally modified. We let the cells do that work with RNA. But it's more than that, because there's really a limitless potential for RNA therapeutics. We can target undruggable targets, heretofore undruggable targets. For example, small molecule therapies really can't do much for children with enzymatic deficiencies. Well, there are programs underway, clinical trials underway, in fact, of RNA for replacing enzymatic deficiencies in children with those problems. As you know, the therapeutic development is rapid by comparison to generating a new chemical entity. So if you want to generate a small molecule drug, you do a screen with your library. Most of the pharmaceutical companies have libraries of millions of compounds. So you have a particular target. It's a particular protein that you're targeting, a particular enzyme or receptor that you're targeting, and you screen your millions of compounds against that target, and then you find maybe 100 hits, and some of those are going to be false hits, and then you have to work out those that are useful. There may be a dozen that have good pharmacokinetic properties, fit the Lipinski rules, et cetera. And then you start testing those, and that testing takes a long period of time. And you have to know if these new chemical entities have genotoxicity or other toxicities. You have to know how they're metabolized. Anyway, it's a long process. And the average small molecule drug, it takes about 15 years at least from the conception of the idea to clinical trials and approval. So with RNA, of course, if it's a particular receptor or enzyme that you're targeting, well, I mean, you can make that. You can just put in the code for that particular protein, generate it. It's simpler to get to the market. The manufacturing process is less expensive. We don't have to worry. I talked about the endogenous post-translational modifications that we allow the cells to do with RNA therapy. And it's a superior in terms of other nucleic acid approaches, DNA-based approaches, viral vectors and the like, because there's no concern about integration into the host genome, and that provides for a simpler regulatory roadmap. Okay, just to give you an idea. Yeah.
Excuse me, John, just because before you move on, we have a quick question here about RNA itself. Is this considered a drug by the FDA, the RNA itself? Mm-hmm.
Yes, it is. It is considered a drug. And we now have 2 drugs that are approved by emergency use authorization by the FDA. Those 2 drugs are the 2 vaccines directed against the spike protein of SARS-CoV-2. So the answer is yes, it's considered a drug. It's regulated by the FDA like a drug. Here's a pipeline for Moderna. This is taken from their website. And you can see that they have about 30 products. And most of these are vaccines, but some are targeting enzymatic deficiencies, and some are modification of cell behavior, like VEGF angiogenic therapy. Moderna was just founded in 2010. Now they already have 30 products that are emerging, and some of those are already in clinical trials, and 1 is approved. They have 3 major pharma partners. They have a $50 billion valuation. So things are moving very quickly with RNA therapeutics. But the question is, why is this happening now? I mean, RNA was discovered, messenger RNA was discovered in 1961. And in fact, the first therapy, at least preclinical proof of concept, was in 1992. So that was over 30, well, it was about 30 years ago. 30 years ago, there was proof of concept that RNA could be therapeutic. In this case, they were injecting messenger RNA encoding vasopressin into animals with this syndrome of inappropriate diuresis due to deficiency of vasopressin. And they could restore normal kidney function in these animals with vasopressin deficiency. But why has it taken 30 years to finally start getting some some drugs? Well, there were hurdles. There are obstacles to overcome. There was this idea that RNA was toxic, and it is, unless you make some modifications. And we'll talk about that, the modified nucleosides that reduce RNA toxicity. And the purification process actually turns out to be even more important. RNA has a short half-life. RNases are ubiquitous. So how do you get over that problem? How do you deliver RNA systemically? How do you get it across the cell cytoplasmic membrane? Now we have carriers to do that, to protect the RNA, to get it across the cytoplasmic membrane, and to allow us for systemic delivery. So we'll talk about that, how those obstacles have been overcome, and how we can help you overcome those obstacles in developing your RNA therapeutic. So the promise of RNA therapeutics has been dramatically shown by the recent approval, emergency use authorization by the FDA for these 2 vaccines, the Pfizer BioNTech vaccine and the Moderna vaccine. The vaccines were amazingly effective. Most vaccines against infectious diseases, if they're if they are more than 50% effective, the FDA will grant approval, can grant approval. But most of the vaccine companies are aiming for something in the range of 70%. But these were 95% effective at preventing COVID-19. And virtually, to my knowledge, and at least the early data confirmed that in these two large trials, that no one that got the vaccine became seriously ill with COVID-19. Now, there were some allergic reactions. We can talk about the cause of those allergic reactions, what's causing that problem, but those anaphylactic reactions are very rare. In the Pfizer phase 3 study, 44,000 patients were vaccinated. The study's still ongoing, but follow-up is still being done. We're learning more from these trials, but the vaccine clearly is effective. And with the Moderna vaccine, it was 30,000 patients that were vaccinated. And again, clearly effective. More details are coming in with longer-term follow-up. So these studies show the future of RNA therapeutics is bright. But what I'd like to say today is that this future is not just the province of big pharma. Everyone on this call can be involved in the future of RNA therapeutics. And one of the ways that can be done is with the help of programs like ours. We want to work with small groups. We want to work with small companies, academic research groups to get their ideas to the clinic. And we produce this platform, RNA manufacturing platform, for ourselves and for the folks in Texas, funded by Cancer Prevention Research Institute of Texas, which charged us with the mission of generating RNA for cancer vaccines and other cancer applications. And we use that funding, about $1 million a year, thanks to the state of Texas and the CPRIT program, to build a manufacturing platform that allows also for clinical grade production. We've developed our own standard operating procedures for the synthesis and the purification and validation of the RNA. And within our institute in the Texas Medical Center, we have other competencies that really will make it easy for us and for other groups that we work with to get to the clinic. We have an RNA biology group and that are doing innovation, that are innovating better forms of RNA constructs that are more stable, that are more translatable, that are more easily translated. We have a comparative medicine program. We were lucky enough to get Tanya Herzog from NIH. She was the chief veterinarian of the veterinary branch. She ran the whole program at Bethesda Comparative Medicine Program. She has tremendous experience in GLP animal studies, small animal studies, large animal studies that you need to get your IND, investigational new drug approval by the FDA. I'll talk about our partner, VGXI, that will make the large batches of RNA. We're at our institute, we make small batches that can support the preclinical studies and also the early clinical trials, first in man trials, and maybe phase 2A. But we have a partner now we have a handshake with them. They're using our processes. They're scaling those up so that we can support the roadmap all the way to FDA approval through phase 2 and 3 trials and commercialization. We have a Center for Bioinformatics and Computational Biology that assists with the development of the RNA sequence, learning, helping us to understand which RNA sequences are more stable, and Nanomedicine group that's helping us with RNA delivery. I'll introduce them in just a moment. And we have regulatory help from our Office of Regulatory Affairs, and we have a GMP director at our Office of Translational Production and Quality, Chris Lincoln. Dr. Lincoln, we were lucky enough to recruit from City of Hope. He built the City of Hope GMP facility from the ground up. And now he's with us, and he's spectacular, helping us move things toward the clinic. So we want to work with research academic groups and also small companies to help you get your products to the clinic. One of the amazing things that we have, I mean, it's just serendipitous that we have a nanomedicine department at our institute. And having them down the hall from us, our RNA therapeutics program is amazing, because we can talk, we can work together, we can develop new nanoparticles together that are more effective at the distribution of our RNA delivery and protection of the RNA. I'll introduce some of these individuals in a moment. If they're on the line, they actually may want to have a chance to talk about what they're doing. Francesca Tarabali is the first one pictured there, and I'll mention some of her work with Christian Boda, developing lipid nanoparticles that are targeting specific sites of inflammation. And the next, Biana Godin has a lot of experience in developing lipid nanoparticles using various lipid moieties to enhance the stability of the lipid nanoparticle and protect the RNA. Christian Boda, I'll talk about his work in a moment, and Francisca Leonard, where she's developing genome editing lipid nanoparticles, and Bruna Cordetti is embedding lipid nanoparticles into collagen scaffolds. Also, we have work going on with silicon particles in which lipid nanoparticles are embedded, the silicon microcarriers for lipid nanoparticles. I don't know if Francesca is on the line, if she wants to raise her hand and talk.
Yes, I'm here, Dr. Cooke.
Wonderful. Francesca, would you like to talk a little bit about how you assemble lipid nanoparticles? And
so we developed a protocol, a standard operating procedure to help people in the Texas Medical Center to prove that their mRNA that they developed is actually functional and it can be delivered. So we are doing two things at the same time. So we help them with the formulation like the Moderna one, very similar to the Moderna one, just to prove that their mRNA is actually work. And we are able to scale up the synthesis till the large animal. And this is the basic synthesis, and we use the nanoassembler to do it. As well as at the same time, we develop different kind of formulation in which, if you remember, if in the audience there are people that follow my seminar back in December, that target specifically some area of inflammation. So we, let's say, work in two different directions, and a direction of serving others to prove the mRNA sequence is actually work and it can be delivered and works in vivo. So we have like a support the work of others, as well as try to develop new innovative formulation, starting from the Moderna one, in order to better target the different site in the body, and to better evolve for the first formulation, and maybe reduce also slight side effects that also Dr. Cooke mentioned from the Moderna formulation.
Francesca, I was talking to Peter Hotez last night. He, as you know, he was on Rachel Maddow last night. He's frequently asked by the media to comment on the current pandemic. He's our friend over at Baylor. I guess he's working with you on a Chagas vaccine.
Yes.
He was very happy with the work you were doing.
Yeah. And this is something that exactly is a proof of what Dr. Cooke was explaining. So these are people from Baylor that came to us and say, we have this sequence is supposed to work for this disease that is called Chagas disease, that is mostly unknown in the North America, but in South America is very detrimental, but they don't have a delivery way. And also they want to eventually scale it up and make it GMP. So they came to us for the translation in GMP of their mRNA to Dr. Cooke, but at the same time to prove in preclinical setting the efficacy of their treatment. And this is what we did with them. And now we are going to move forward.
Thanks, Francesca. You know, on the next slide, I think you've talked about the work of Asaf and Christian Boda and Asaf Zinger, who work with you to develop this biomimetic approach to treat cardiovascular disease. But I think we have some people that may not have heard that story, so maybe you could just say a few words about about this approach. And
yes, if you remember, we are expert in what are called biomimetic platforms. So there are biomimetic nanoparticles that use the cells tropism, in our case, leucosome, in other case, other kind of cell proper tropism to target inflammation. And this was excellently proved from Christian Boada and Asaf in this circulation research paper, in which we use our platform leucosome, in which we get the membrane from the leukocytes, and we use the tropism that we got from this membrane to target inflamed area. In this case, we try to basically embed these particles inside the normal formulation of liposome. And this platform also is being loaded by rapamycin, because this platform is very useful to embed both hydrophilic or hydrophobic drugs to target plaque in a arteriosclerotic model in mice. And by the way, we are moving forward in rabbit together with Dr. Cooke, and we scale up this platform as well.
It was wonderful to work with you on that, Francesca. That was a lot of fun, and it was quite impressive how the leucosomes, which are essentially lipid nanoparticles with the embedded white blood cell proteins, they track to sites of inflammation, and they deliver whatever you put inside of them. And in this case, we didn't put RNA in. We could have, but in this case, it was rapamycin, which is a potent inhibitor of proliferation, and it did. It had inhibited the proliferation of white blood cells, macrophage specifically, in the atherosclerotic areas of the ApoE deficient hypercholesterolemic animal. Beautiful work published in Circulation Research last year. Thanks, Francesca. We also have a RNA biology group led by Dan Kiss, who helps us with enhancing RNA stability and translation. We do a lot of things that others do, but what we do is we do it in a way that allows us to synthesize clinical grade RNA. But we modify the RNA bases to reduce immunogenicity. We optimize UTR sequences to avoid microRNA binding sites. We tailor the RNA sequence to maximize codon optimality. And Dan, I'll show one slide. He's circularizing RNA to add stability. And then also we can enhance, Dan has approaches to enhance translation. There's a translational ramp that you should be aware of. There's specific amino acids in the early codons 3 to 5 that can increase protein yields if you have the correct translational ramp. You can maximize codon optimality. And then structure-free 5' UTR seems to be important in maximizing the efficiency of pre-initiation complexes and translation. Here's Dan in the lab, proudly holding his first vial of circularized RNA vaccine against the spike protein. And sometime he might talk about this work to you, but just briefly, it's much more stable on the shelf. As you guys know, the RNA vaccines have a problem with cold chain, and our preliminary evidence suggests that we won't have the same kind of problem with the circular RNA. And we've developed some lyophilization processes, and the RNA that's lyophilized is stable, absolutely stable for 3 months, and still pretty good at 6 months in the fridge. And you know, the Moderna vaccine can only last 5 days in the fridge before it has to be tossed. In vivo, the circularized RNA vaccine is much more stable than linear constructs. Linear constructs, in general, endogenous messenger RNA, 90% of it lasts from 2 minutes to 2 hours. The linear constructs for the RNA vaccine are really not any different. The protein is produced lasts longer, of course. But the RNA itself is gone rather quickly because of endogenous endonucleases. But the circular RNA, of course, it's immune to the exonucleases, excuse me, because it doesn't have a 3' or 5' end. So it's much more stable in vivo as well. So at least in cell culture studies, it's stable for days as opposed to minutes to hours. So that's a new, exciting new approach. We have up on the 12th floor of our facility, we have clean rooms for GMP synthesis. Down on the 10th floor, we generate the plasmids, we generate research constructs for investigators, just research grade RNA on the 10th floor. So we can help you generate the plasmid vector design. We can do the IVT synthesis, and also purification turns out to be absolutely critical for the biological, for the cell viability and tolerance of the RNA. Here's Roman Sokoverson, who's our scientific director in the lab. In this case, he's doing HPLC purification. For larger batches, we've developed a different purification approach that allows for large batch purification. There's a piece of data there from Carrico's study. Catalin Carrico is one of the pioneers of RNA therapeutics, and she was the first to show that modified nucleosides could reduce innate immune activation by the exogenous RNA, which was important for cell viability. This heat map shows the upregulation of inflammatory cytokines and chemokines by the RNA. And on the right side is very strong activation of these inflammatory cytokines, which you really don't want. Can actually lead to the death of the cells that are being treated. And when she added pseudouridine or 5-methylcytosine, she saw a slight reduction in the activation of innate immunity, and in some cases, an abrogation of the particular inflammatory cytokine that was being upregulated. But in the 3 left panels, you can see that when HPLC purification was added, that had an even more dramatic effect. So when people talk about RNA activating innate immunity, they talk about the modified nucleosides as playing a major role in reducing innate immune activation. But you can see in this slide from her paper that the purification actually is even more important. Getting rid of the abortive transcripts and RNA DNA hybrids is more important to improve the biological tolerance of the RNA. I mentioned that, so we can help you get to, through the preclinical studies, GLP studies, we can give you clinical grade RNA for that, and we can help you with first in man, maybe even phase 2A trials. But when it gets to phase 2B, phase 3, where you need larger batches of RNA, and certainly for commercialization, that's really not our sweet spot. We have joined up with a CMO in the Woodlands, which has a lot of experience. VGXI has a lot of experience in DNA vaccines. So they licensed our proprietary processes for synthesis and purification and validation of RNA therapeutics, and are scaling up now, so that we can provide a full assembly line, all the way from innovation and development through preclinical studies, through phase 1, 2, 3, and commercialization. This company really believes that nucleic acid therapies are the way to go. They just opened up, they just broke ground on a new facility, 44-acre site in Conroe, outside of Houston. The initial facility is 125,000 square feet, but their longer term vision for this site is a 1 million square foot complex for manufacturing. So I think a lot of people recognize that RNA therapeutics is the future and are investing in it. Just some examples of how we can help you get to the clinic. This was work we did for Rongfu Wang when he was here in Houston. He's now at UCLA. We generated for him an RNA vaccine against melanoma. So he has a nice model for melanoma in mice. And you see in the picture on the left, when these animals are injected with melanoma, it metastasizes to the lung. You can see the lung is speckled with metastases. But the metastasis is reduced by the vaccines. Now, one thing that was very interesting that came out of this is the vaccine we made for him, the mouse tyrosinase, which is a melanogenic protein, the TRP2. That vaccine worked in the murine form of it worked in mice, but when we made the human form of it, because we're planning on getting to clinical trials, he's planning on getting into clinical trials with us. He made a human form. We made it for him. He injected that. It actually worked better in the mice than the mouse TRP2. So it's probably, it's close enough to, there's enough conservation of sequence that it resembles, of course, it resembles mouse TRP2, but it's different enough that it just triggers a stronger immune response, which makes us think, well, maybe in the human trials, we should be using the mouse TRP2. But that will be up to his company to figure out, but we could certainly help him test those ideas. This is work from Johnny Chen at Baylor. He had this interesting finding that exist, the long non-coding RNA exist, can suppress pancreatic cancer. And that's shown in the first tumor weight inhibition with his exist long non-coding RNA vaccine. He has his own lipid nanoparticle that he's developed, polymeric particle that he's developed to carry the RNA. But using the exist RNA that we made for him in his lipid nanoparticle, he had rather dramatic effects on pancreatic cancer in his murine model, shown on the right. Just some examples of how we can help academic investigators move their work to the clinic. Here's where a hospital-based program can actually be more effective than a big pharma. I do believe that personalized cancer vaccines can best be addressed by local groups, regional groups for their patients. And it does require a system, and we can support this kind of work. You need a computational pipeline for generating a personalized cancer vaccine. And this is a nice paper from Hundal and Martis in 2019. And the idea is that when the patient has their operation to remove the tumor, you've got, you can do a sequencing of that tumor and compare that to a sequence of normal cells in that patient. And, pick out those that are different, the cancer mutations. And then with your computational pipeline, determine which of those are more likely to be bound by the major histocompatibility complex in that patient. And you can also do screening of the RNA being produced by that tumor to see if those peptides are actually being produced. And that kind of screening can help you to design your nucleic acid-based vaccine. And on the right, they show, well, you can use a plasmid DNA or you can use RNA vaccine. And they talk about in this article about how nucleic acid-based vaccines are much more elegant and rapid to the clinic than our recombinant proteins, which, as I mentioned before, have more problems with manufacturing. This is something we're excited about. This is an in-house program. When I was at Stanford, finishing up there before I came to Houston, we did some work on RNA telomerase. And what I'm showing you here is population doublings on the y-axis and time on the x-axis. These are fibroblasts that were brought to a point of replicative senescence. Human fibroblasts start slowing down at about 50 population doublings. With 1, 2, or 3 treatments of RNA telomerase, we were able to give the cells extra shelf life. And even after they start to plateau again in terms of their cell doubling, we can treat them again and they have more replicative capacity. Not too big a surprise that exogenous telomerase can extend the telomere and improve replicative capacity. But what was surprising, what allowed us to get intellectual property on this idea, is that a single treatment could have an effect, and 2 or 3 even more effect. And we've done a lot of work since with progeria patients, cells, and other human-aged cells to show that this approach actually works. It can increase replicative senescence, also improve function of the cells. Here's an example of CAR T cell therapy, which could be enhanced by this hTERT mRNA. One of the problems with CAR T cell therapy is in some patients, you just can't get enough CAR T cells. You're expanding the T cells ex vivo. After you've introduced the chimeric antigen receptor genetically into the patient's T cells, you need to expand them ex vivo. And you need about 500 million billion cells to have an effective therapeutic. But some older folks particularly can't get that far. The cells poop out during that expansion. Well, in the second panel there in B, you can see again population doubling over time, and you can see that the treatment with human telomerase can markedly improve replicative capacity of these cells. When these cells are injected into mice, they do better. Mice that have been, in this case, the CAR T was against CD19. It was against B cell lymphoma. And in these immunodeficient animals treated with the human, are exposed to the human B cell lymphoma, tumors would grow and kill the mice without any treatment. And with treatment with CAR T, they had longer lifespan, but with the TERT-treated CAR T cells, they did even better. So we're very excited to announce this new collaboration that we have with Avita to improve their skin product with our RNA telomerase. They have a wonderful product for burn patients. What we normally do with a burn patient is we take a patch of skin from an unburned area, we mesh it to expand it, 3, 4, 5 fold, and then we place that over the burn site. Well, what the patient is left with, that does work. What the patient is left with then is, when that heals, is they're left with kind of a meshwork on their face. So it's not an appealing cosmetic result. What they do instead is they take the unburned skin and from the donor site, and they disaggregate it and spray it on the surface of the burned site. And you can see that the cosmetic result is much, much better. This works great in younger folks with burn, but not so much older patients. And so what they want us to do is see if we can improve their therapeutic with RNA telomerase. So we've started that project. The RNA telomerase is encapsulated in LNPs that we're making using the PNI device, the nanoassembler. And we're just now getting started in seeing optimizing that approach. So anyway, what I'd like to end with is by saying the future of RNA therapeutics is not just big pharma. I think everyone on this call can participate. We've developed the first hospital-based RNA therapeutics program. We want to help other academic groups and startups that have a deep fund of knowledge about their idea. We want to help them develop their RNA therapeutic. We have in-house RNA biology expertise to help you to improve the construct, and we can generate proprietary processes. We have the CGMP, the expertise, and the facility for clinical grade RNA production, as well as clinical grade nanoparticle production. I don't know if I mentioned, we have the PNI device, the nanoassembler, up in our GMP facility also, so we can make clinical grade nanoparticles. And we have the animal models and the GLP expertise to do the testing for FDA, for the IND, and internal regulatory guidance to help us and to help you. We have a phase 1 unit for first-in-man clinical trials. We have a large hospital system with the capacity for doing larger clinical trials, phase 2, phase 3. We can't make RNA for large batches at our facility, so we have this corporate partnership with VGXI to help us. They'll make the large batches for those later trials. And we're generating our own IP and products, and would love to work with people on this call. We want to make our hospital a destination hospital for RNA-mediated therapy. You can do that for your hospital as well, or you can work with us. We'll certainly would love to help you get there. So anyway, we have this RNA therapeutics program at Houston Methodist Hospital. It's housed within our Center for Cardiovascular Regeneration, some of the folks shown here, and our collaborators and funding. Thanks very much for the opportunity to talk, Kimberly, and I'll take some questions if there are any. Oh, one last thing. We have faculty positions open in RNA biology and chemistry. Just contact me, and love to look at your application if you're interested to work with us.
Well, thank you again to the team at Houston Methodist Research Institute. There's a lot of exciting research being done, and we're thrilled to partner with you. We really were excited to have you kick off our webinar series. We're actually continuing the Texan theme. Next month will be Dr. Ghosh from University of Texas at Austin will be our speaker. So if you haven't registered, we encourage you to register for that as well. Hope everybody has had a great start to their new year, and we look forward to working with you in 2021. Have a great day, everyone.
Bye-bye.
Bye-bye.
Thank you. Bye-bye.